So that's kind of why we're talking about cancer projects. Whatever project you would like to do is fine. It's for you to be able to practice some of the methods and the ideas that we discussed. And in the context of this program, we're going to be discussing projects that are only related to cancer. We have another program that's going to start that's going to be focused on infectious diseases. So we can leave that for there, but it's up to you, whichever one you would like to do. But the idea that I am going to discuss here, right, as a project idea that I propose based on some of the topics that we discussed up until now, is here I have a lot of data. So let's look a little bit at what kind of samples do we have. 3,678 patients with breast cancer were studied. For 405 tumors, a comprehensive multi-rater histopathological evaluation was performed. Using RNA-seq data, single gene classifiers and multi-gene classifiers were trained on consensus histopathology labels. Trained classifiers were tested on a prospective population-based series of 3,273 breast cancers that included a median follow-up of 52 months. And results were evaluated by agreement statistics in Kaplan, Mayer, and Cox survival analyses. So we'll talk a lot about what these things mean today and how do we translate into specific practical steps that we can do based on the analyses that we discussed in the context of this program. So now you can see that there is actually 3 different data sets here. And so those are some of the links that I would add into my form, right? So that is the link to the raw data. And then what is the reference publication? Again, that's a nice description, but where is that publication? So the publication is right here. In case some of you want to take a look, here's the publication. So cross-comparison and prognostic assessment of breast cancer multi-gene signatures in large population-based contemporary clinical series. This was published recently in 2019, and you can see how this topic here, sorry, I pasted the link to only ViewChat, so let me post it to everyone. So you can take a look, right? So multi-gene expression signatures is what we will talk about today, and we'll talk about different things that are related to risk prediction, but also related to selection of treatment in the context of breast cancer. So this is a project that, again, I selected, and I am going to try and do some of the analysis together with you so that you can also think about some of your own project ideas as we go through the remainder of the course. So are there any questions about how I filled out this form, or is it pretty clear to everyone how to fill this out? Yes, sure. I would love to pair up with anyone who wants to work on this data set. Or if you would like to pair up with another participant of the program, that would also be great. We're actually trying to get everyone to complete their profiles as much as possible so that we can review. There's actually a lot of really interesting people that join these programs. So I'm hoping that all of you can learn to interact with each other during this time, because we have, so let's actually take a look. I think I pointed it out. So here's the link with all of the people. I was impressed with some of your profiles that were with all the descriptions. You can actually connect with people. A lot of people put their links to LinkedIn or Twitter. You can read about them. So I think, that would be great if we could create groups of people that are interested in common projects. And obviously, there's a lot of interesting questions that could be proposed by people that have that background and are learning how to use some of these tools. So again, happy to pair with anyone who's interested. The way to facilitate this process, again, is to fill out the form. Obviously, it's not restricted for people in the program. You can do it with whoever you want. The thing is that in the context of the program, we're going to be having these regular discussions. We can set up, one-on-one meetings or team-specific meetings. And also we can, obviously discuss data sets in the context of the platform. A lot of the analyses are going to be much more reproducible. Again, you're welcome to do whatever you want with this, but, in the program, it's going to have a lot more context. Another comment from Iftikhar, my network system was down from 2 weeks. I could not follow the classes regularly. I'm still to complete the courses. Yeah, obviously. So if you still are working on the courses, please continue to do that. There are also some projects here related to cancer that you can take a look at, and we'll go through one of those examples today. So if anyone has any other questions about how to fill out this form, the purpose of this form, the purpose of this stage of the program, right? Again, we are working on independent projects. Your data set, a public domain data set, a data set associated with a question is where we want to get everyone to be. And then we can have these more informed discussions about what are the analytical steps and how to get that project to a full analysis during this time. Is there a deadline? There's not a deadline. This is up to you. The program itself allocates about a month for working on the project. So if during the next month you can complete some of the analysis and get it to a point where you feel comfortable presenting it, that's great. If not, you can return to this at any point in time where you have more time, right? So this is not going to be required of you, but we do encourage that because we believe that working on your own data set will actually bring a lot of these ideas to life, and you'll be able to really think critically about the methods and the data that we are looking at. Okay, so Asma here is adding a comment. Resectable breast cancers could be good for FFPE, DNA, RNA extraction, which they used in this Nature article. Great, because from biopsies, we couldn't get good yield DNA. Resection tumors are good too for wet lab, which will give us about 0 artifactual sequencing results. Okay, so a comment, again, I probably feel like a lot of you have a lot of expertise in the wet lab perhaps, or working with breast cancer patients, etc. So, I'm glad that a lot of you might bring more of that perspective to these projects, right? So I think by interacting with each other, we can learn from each other, learn from how the samples are prepared, what do they really represent, and then from the data analysis, how do we apply some of these tools to the data sets that we have access to, and what types of results are we expecting to get. So thank you for that comment. Anyone else wants to add anything to this specific project, or have us discuss any other ideas or questions that you might have for this form? Okay, so if not, I want to, first of all, review a little bit of what we did last time and make sure that as you remember last time, we were not able to run a differential gene expression analysis, and we were not able to run factor regression analysis with the TCGA data. And we were also not able to load the SRA links. So that has been fixed. And so now I'm going to show you how to do that. So in case you would like to do that for your project, you'll know how to do that. So I'm going to close this for now. Okay, so again, if you have any other questions, you can put them there. So we talked about different data sources and databases, right? So we talked about NCBI, we talked about TCGA, right? So TCGA will provide us with prepared data, processed data, data that already is in a table format, and similar data we can get from Geo. We also have NCBI, where we have both compressed raw data, so SRA, but we also have some processed data, so gene expression as well, you can find on NCBI. And then on NCI and Cosmic, we have references for different positions, annotation of those positions, etc. So we are talking about some of these reference databases. And we talked about how to think about project preparation. So we have here these 6 steps outlined for you to kind of think through the project that you are thinking of, framing the problem. What does that really mean? What is the question, right? What is the question that you want to answer? Step 2, collect the raw data needed for your problem. We talked a lot about how process data can bring a lot of, on one hand, it simplifies the analysis. On another hand, then you need to find out how exactly was it prepared. Then we need to process the data, prepare it, structure it, explore it to identify any variation that is unwanted as opposed to wanted, and really start looking at the different types of information that we have about the samples, right? The clinical data, the phenotypic data, the expression data, or the genomic data, etc. Perform in-depth analysis and then communicate the results of the analysis, right? So some of these steps is what we're going through during this time. And so last time we talked about TCGA patient data. We downloaded some data. We looked at some of those aspects of annotation. So what are these samples and what types of clinical information we have about those samples? And then looked at some of the process data that we downloaded from Firebrowse. And again, sorry that some of you had those problems. But then we talked a little bit about data mining, and we talked about how we could, for example, apply principal component analysis to ask specific questions, right? So does anyone remember what question we were asking as we did this principal component analysis from last time? All right, great. So tumors in breast versus liver. Okay. So again, the question is, do you remember where we finished off last time? So we can kind of move forward with that idea. And so Asma, thank you. How can we further do this analysis of differential gene expression? So anyone else? So what we were trying to ask about this data, right? So this is principal component analysis, right? So what was the question? It shows us difference between different types of samples. But what were we trying to ask now? Percentage of difference, yes. So we saw how there was a difference between, so let's just explain what this chart shows, a difference between liver cancer, right? So this is liver tissue. Some of these are tumor samples. Some of these are normal samples. And then we had breast tissue. And here we had, again, normal tissue and tumor tissue. Right. And so what we saw, so our first step was exploratory, right? We were like, okay, let's explore this. What can we learn? What does the data actually tell us? And the data told us what? What was the more significant difference? Yeah, what was the more significant difference? Was it between tissue types or was it between normal versus tumor? Tissue types, exactly. So we saw from this exploratory analysis that the major difference is between tissue types. So here we have liver cancer, here we have breast cancer. How did we identify those differences? We did principal component analysis, right? So we did principal component analysis, and now you see the same chart, but with those percentages. So what is the bigger explanatory principal component here, principal component 1, right? And so PC1 has about 65% of explanatory power for this analysis, and these samples here are the different tissues. And so the next question that we want to ask once we've explored and identified that that is so, we can ask a question, well, how can we characterize that difference? And the way we can characterize that difference is by looking at specific genes that demonstrate this difference, right? Because just knowing that there's a difference based on RNA-seq doesn't really tell us anything, right? Doesn't tell us anything specific about biology or functionality of the different processes that are happening associated with those tissues or with tumor versus normal. So let's take a look now at the gene level data. So this is gene level data for this gene, EGFR. Again, this is from Firebrowse, just like we did last time. And if you just search here for EGFR, you get this plot. So based on this plot, could you tell me what is the difference in tumor versus normal in breast cancer for EGFR? So what is the difference in the expression of EGFR in breast cancer, specifically for breast cancer data set? So it's downregulated. Let's be more specific. Downregulated in tumor. Okay, so let's see, how did you find that, Zainab? Where should I look? On x-axis, y-axis? Where am I looking to find that information that you pointed out? Okay, so what is y here? Y is our SEM, right? And how do I find the breast cancer data? BRCA. Okay, thank you. So I will look at BRCA, which is right here, right? And here I see tumor and normal, right? So here's the tumor data, here's the normal data. Now let's find the liver cancer. Where's the liver cancer? What would I be looking for to find the liver cancer? LIHC, thank you. So here's LIHC, right? And so what do I see here? That both BRCA, right? This is downregulated in tumor, upregulated in normal. LIHC, the same thing, downregulated in tumor, upregulated in normal. And so that's why I don't really see a difference between the tumor and normal, right, as significantly as I see between those two, because here you can see the difference is much bigger. It's about 2, which is significant, and here it's insignificant, so it's about less than 1, it looks like, right? So if it's less than 1 here, then that difference between tumor and normal is, if we just took this one gene, right, would not be as significant. So now let's take a look. For some reason, this. So here we can see another example, MMP11, right? And so now what we want to do is we want to ask this question, right? How do we find all of the genes that have a difference? So some are going to be upregulated in tumor, downregulated in normal. Some of them are going to be the other way around. So how do we do that? We do that with differential gene expression. So just like we did last time, let's go back to the platform. Okay, we aren't going to get to the new data that we have for today, still kind of trying to finish up what we did last time. So let's try and do this quickly. We have here differential expression, right? Now we need to upload the data. So we go to File, Upload. Let's upload the data. So last time we talked about you had the data from last time, right? And so that was we had here the normalized data already, right? So let's just check this data before we just put it in. What are we checking for? So we're checking for the column names. As you remember last time, here you have gene ID. What you want to have is just ID like this in the first column. So let's choose. And then we have group selection. And so now you can see all of these, right? So what do I put in which group? What would you want to compare? You think about that, right? I'm going to just compare, for example, all of my breast cancer tissue from all of my liver tissue. So I would take all of the BRCA ones and put them in here, and all of my liver ones and put them in here. And now I can just connect, for example, DE-seq2. Here you select which database. So we're going to use the human database and run this pipeline. So what will this provide us with as an output? Who can tell me what are we expecting from this pipeline? What will we get? So who wants to tell me what are we expecting to see as a result? Okay, so differential expressed genes between liver and breast there. So the up-down regulated genes, right? So we're going to find all of the genes that have a difference in expression between breast and between liver tissues, no matter whether they're tumor and normal. Are we introducing some noise in this analysis? What do you think? Is there going to be some noise in the analysis that we're doing? Yeah, so please explain. What do you think the noise would be? Okay, so several patients. Right, exactly. So there's not going to be any separation between normal and tumor, right? So tumors still, even though might not be the biggest difference in the samples that we see here, but it's still going to be a difference. And so we're not going to account for that difference. So the limitations here are going to be significant. Nevertheless, let's take a look at the result. So the result here, we will look at this file at the end, so DE-seq2. And what will that result in? By the way, all of this information is in is going to be in yes, is going to be in transcriptomics 2. And there's going to be an explanation on how to run this and what these outputs mean, but let's take a look. So here we have the results. As you remember, there is going to be a p-adjusted value. What is this p-value adjusted for? Who remembers? Multiple testing, exactly. So what is that called? No, it's called false discovery rate. So it is a problem of multiple testing. We have here thousands and thousands of genes. Just by chance, you're bound to find some having significant differences. And so what we want to do is we want to adjust by counting how many total observations we have, how many genes we have, and adjusting for false discovery rate. So let's take a look. What would I do here? I would want to select for less than 0.05. This is already adjusted, so I don't have to, take anything else. And now let's just check how many do I get in the end. How many do I get in the end? Let's just copy this over here. Okay, and let's take a look. How many differentially expressed genes do I have? Okay, so I have close to 7,000 differentially expressed genes. That is between liver and breast cancer tissue, right? Not all of them, however, have the same amount, right? So let's take a look at the differences between log 2 fold change. So this is logarithm of the fold change. And so what does this fold change represent? This is by how much, right? So as we saw over here, right, the different examples that we were looking at, in the breast cancer, the fold is big. In the liver cancer, the fold is small. So this is what we're trying to look at right now, is the fold change. So let's go in here. And now what do we want to select? So some of them are going to be liver cancer, or sorry, liver tissue upregulated, breast tissue downregulated. Some of them are going to be the other way around. So that's going to be the negative and the positive values right here. So let's select, let's do a filter. And I think 1.5 or perhaps even 2 is going to be a good threshold. So we can say greater than 2 or less than -2. So let's see how many do we get now. So we have about 3,600, about half of what we had, 3,600 genes that are differentially expressed with a significance of log 2 fold change of 2 or more than 2, right? And now we can see how many genes we have and what they are. And so the next thing that we can do is we can annotate them to understand their functionality. But we already said that the problem with this analysis is that we actually don't know that we are not accounting for the noise that comes from multiple factors playing a role on gene expression, right? So how do we address that challenge? The way to address that challenge is to actually look not just at two conditions that we have here, right? And find genes that have perhaps multiple variations, but nevertheless show some kind of a difference between the two groups. What we want to look at is factor regression analysis, where we are measuring the relationship between independent and dependent variables based on regression or relationship between them. So how do we do that? Again, this is something that we do in transcriptomics too. So I suggest for everyone to review that. How do we do that? Let's go back on the platform and quickly do that. So we go right here to factor analysis, and please stop me if anyone has any questions about what I'm doing. Here, the way you set up the pipeline is a little bit different. You first have to get to factor analysis. So we click on factor analysis. And we can say if your samples are not normalized, you can perform different type of normalization, either within groups that you will prepare here, or you can do for the whole data set, so quantile normalization for the whole data set, or no need. So let's just do no need for now. Now, how many factors do you think we have here? Who can say how many factors do we have? 4 factors. Okay, so what are the 4 factors? What are the 4 factors, do you think? Okay. Breast normal, breast tumor, liver normal, liver tumor. Okay, so that's pretty close, but not exactly, because we only have two factors here, and each factor has two levels. So we actually have the tumor versus normal, that's factor one, and then liver versus breast. And what those variations are called are levels, right? Because for each factor, we will have two levels, and in total, indeed, we will have four. So let's set up 2 and 2. We'll now load our data, so the exact same data, this one. Okay, and now we can check, okay, and drag those files into the appropriate factors and levels. So exactly like what Elton mentioned here, we have breast normal, breast tumor, liver normal, liver tumor. Let's drag them in an appropriate way. So what is A1? Let's say that our first factor is going to be tissue because it's a more significant factor, right? So we want to say A1, A1. So that's going to be breast, breast, just like you noted here. So which one do I take? So these are not tumor, 3, these are tumor, right? So I'm going to take, just like he said, so I'll take A1, B1, so these are the not tumor, these are the tumor, right? And then my factor 2 is going to be liver, so let's take the liver normal, and liver tumor. So what I'm going to be able to do here with this kind of analysis is understand the variation between these different factors, find how select for in my results, how different genes, so find genes that are responding to both only the tissue type or only tumor versus normal, or perhaps some interaction of both, right? So, and that would be the interesting find here as well. So if I am able to find genes that resemble some difference between tissues, but at the same time, when there's a cancer process involved, they have some unexpected high or down regulation as a result of that change. So those would be some interesting questions. This is how you set up the pipeline. What is going to be in the pipeline output? In the pipeline output, you will have the results of each factor influence on the different settings that we set up. So here you will be able to filter these columns and find which kinds of genes. Genes, for example, that have only factor A, or genes that have different types of variation, right? So this is based on the PDX project. If you want to practice how to do this, again, this is going to be covered extensively in transcriptomics 2. So we already talked about how to set it up for this analysis, and here's just an example of some of the results. So some of the results, what do we see here? We see here upregulation, right? So here you have tumor tissue, non-tumor, right? So you have some up, down, right? So this is consistent with tumor versus not tumor, tumor versus not tumor. But at the same time, right, there's some interaction of factors, because with the breast tumor and with the liver tumor, there's also a significant difference. So those are some of the genes that you will be able to find, and those are some of the variations that factor regression analysis can display. Okay, so any questions about this so far? This is kind of a little bit of a continuation from what we did last time. Okay, so let me know if you have any questions. I'd be happy to answer them about this. So another challenge that we get to once we get to more and more complex data sets is the challenge of multiple groups or multiple subgroups that we have in these complex data sets. So we started talking about interaction between genes. So we cannot find direct interaction between genes by using factor regression analysis. Factor regression analysis allows us to find interaction between factors in sample, right? And then we have um, influence of that factor on gene expression. But we will talk about network analysis. Network analysis allows us to actually look at some correlation between genes and how those could be connected into nodes that are interconnected, and that would be interaction between genes, right? But the same could be done in multiple ways. We can do it by annotating pathways, that's interaction between genes. We can do it, so that's biology driven. We can do it analysis wise, that's for example network analysis, or some kind of clustering of genes. We can do almost the same thing there. But here we're specifically focused on trying to find correlation between phenotype and expression of genes. Okay, another question here from Niche. If we do t-test, will the results be the same? Okay, so actually the results are going to be very similar. What is t-test? T-test is difference between average per group, and you can compare groups, right? And then it will help you select statistically significant ones from not statistically significant. The problem with t-test is that you then manually also have to add the account for the variability within groups, which t-test does not consider, and also false discovery rate. So differential gene expression allows you to do all of those things and uses the same statistical principles, but accounts for different types of variation and makes that variation have an impact on the p-values that in the end you get in your output. In terms of factor regression analysis, it's different from t-test because it's not looking at just two groups, right? It's looking at multiple different factors using regression. So you would have to do multiple t-tests to find the same thing. All right, so now let's focus on the next challenge. The next challenge is having multiple subgroups and a lot of variation in your data. So let's talk about this cell line project. This is taken from Transcriptomics 3. So please make sure that maybe by next time, all of you can try and go through Transcriptomics 3. It talks about machine learning, clustering, classification. We will use these in our next session on liver cancer as well, because we will be able to play around with a whole variety of different factors, not having to follow this strict setup, right? It's going to be much more flexible. And the differences that we will find are going to be different types of differences, not described by p-values and fold change, but described by more subtle variation. So we'll talk a lot more about that in the next session. But Transcriptomics 3 is a course that allows you to look at a variety of different data mining and classification methods to help you extract the most of your data, studying relationships between sample metadata, so what you know about sample phenotype, and the type of data that you have in already structured, already prepared in a table. So again, how do these transcriptomics courses connect? We kind of go through processing the data and seeing what is in the reads and how do we structure that data, to then discussing statistical methods like we did today with t-test and with factor regression analysis, and then going to machine learning and classification, which is in Transcriptomics 3. And so in Transcriptomics 3, we start talking about this new data set. It's modeling precision medicine, cancer precision medicine. It talks about breast cancer cell lines. We saw that a similar approach is taken in the paper that we started off today from Nature. But essentially what the authors of this paper did is they tried to identify specific responses to different drugs on one hand, and on the other hand, use molecular classification, so separate your samples into different subtypes, and then find a relationship between them. And the question was, could we find a relationship between molecular classification and response to treatment, and design an artificial intelligence workflow where we can have a doctor prescribe the right treatment based on molecular data and based on training data from high throughput testing of different molecular treatments. So let's kind of break down this process into more simple terms. So as you know, we have this concept of clinical subtype. So here you see a mammogram. In this mammogram, we have a tumor. And one of the questions that we're trying to ask is how to characterize this tumor. And so when we think about how to characterize this tumor, we can talk about which tissue it's stemming from, right? So we saw that there are a lot of differences between tissues. And so if we knew what tissue this is stemming from, we would be able to define maybe how this tissue is changing as a result of being a tumor. The problem is that it's not always easy to determine what tissue this is from. And as cancer progresses, it becomes infiltrating, right? So we can see here invasive ductal carcinoma, meaning that it stops being characteristic of what the tissue is and expands beyond that tissue, losing its differentiation. The other process that we can use is by looking at expression of proteins or protein markers associated with different subtypes. So we saw in the beginning, right, how we have also an evolution of how we think about these different subtypes of breast cancer. It used to be 4, now it's 5. Those 5 are also changing. And so one of the ways that we can look at all of this is by looking at expression of genes. And expression of genes, so here you see a molecular clinical panel called the PAM50 panel. And the PAM50 panel divides all of the samples based on 50 genes. Not all of them are actually playing a role in the classification, only 35 or 36. The other ones are called housekeeping genes, but what they provide us is with a nice separation between these different subtypes, 5 different subtypes of breast cancers. And so what you can see here are these patterns, and these patterns are essentially what separate them, but they also have a clinical purpose. So what is their clinical purpose? Clinically, we can use these panels for prediction of survival and recurrence. So here you can see some of those things that we started talking about in the beginning, Kaplan-Meier curves that show separation between 3 different groups, high risk, medium risk, and low risk. And you can see how those are referred to in terms of recurrence, risk of recurrence, and risk and rate of survival. So rate of recurrence, rate of survival. And you can see that there are multiple panels. Actually, this is a comparison between PAM50 and Oncotype DX, 2 clinical panels that are being used for this kind of classification. But you can also use this kind of approach to select treatment. So because of risk of developing, for example, recurrence or survival, you might want to choose more aggressive, less aggressive treatments, and also identify treatments that might be treatments done in stages, right? So for example, radiotherapy before chemotherapy or before surgical therapy. So different types of treatment selections, right, will play a role. This kind of data will play a role on selecting those treatments. Another type of treatments is targeted cancer therapy. And so a good example of targeted cancer therapy is Herceptin. Herceptin is a drug that targets the HER2 receptor. And so because it blocks that receptor, the HER2 receptor is a signaling pathway that signals for the cell to replicate. And if you block that receptor with Herceptin, it stops that uncontrolled proliferation, which is a signature of cancer. And so if we think of the same idea, right, now we know that the expression of a specific gene leads to appearance or proliferation of these receptors. We can block those receptors. So if we find that this receptor is amplified, we can then select for a targeted treatment to stop that process. This is just one example. There are multiple examples now like that. So what they did in this paper is they took specific molecular profiles, right? So they designed a process, a data mining process where you first identify genes responsible for separation to different groups. Once you find those, you can then find associations with the drug data, so response to different treatments. And they took here different drugs that are either in clinical trials or already on the market, chemotherapy, targeted drugs, etc., and try to find associations that would help us predict response to drugs based on molecular profiling. So I think a very fascinating study. Similar studies now exist in other types of tissues and other, not just breast cancer, other types of cancers, but also with improved accuracy. And so one of those that we looked at today in the very beginning that I proposed as a study is actually a very similar approach, right? So here we have immunohistochemistry that looks at markers of tissues. Based on those markers, we know how to select different treatment strategies. Could we use RNA-seq and develop multi-gene signatures, which is what we will do in today's session, and identify how they help us separate this into very specific subtypes. So where do we find the raw data? The raw data is found on NCBI, and so I'm going to use this example to help us find the raw data and load it onto the platform, because I was trying to show that last time and it didn't work, but we fixed that. So we can take this link right here. I'm going to just paste it here, okay? And let's, again, sorry, pasted it for everyone. Okay, so what we'll do is we'll find this data set. Let's find it. How do we get this data onto the platform? So how many samples do we have here? We have 56 SRA experiments. So to download 56 of these files, you know, this is going to be a huge data set. So instead, what we can do is send to file, and here we can have, sorry, we first need to go to SRA experiments, send to file, and here run info. Then we have to find that file that we downloaded. Let's open it in Excel and see what's inside. So what we have inside are the runs. So these are going to be our sample names. And also we have a link to the download. So you can see that this corresponds to this right here. It tells you that this is transcriptomic data, paired end sequencing, right? And then here you can see that this is Homo sapiens, so this is human data, and that this is public consent because this is going to be cell line data. So this is RNA-seq, and those are the samples. So let's just save this as a TXT. So instead of CSV, let's use tab delimited text. We'll save that. And now let's upload it on the platform. We'll go to RNA-seq. Remember, was this single end or paired end? Just making sure that you're all paying attention. Paired. Okay, so asthma. Remember that where did we see that this is paired? We saw right here library layout, right? Paired, right? So we got to make sure that we select the right settings. So we change this to PE. Now these are going to be FASTQ files that are packaged inside SRA. So we keep this as FASTQ, and here we'll use the human assembly 38. So let's, yeah. So let's upload, and we use this NCBI run table upload. So let's add files. Tried showing that to you guys last time, did not work. So it should work now. Okay, so now we uploaded this group selection, and there we go. So now we have all of these samples that are going to be automatically downloaded from SRA. We don't need any groups right now. And now we can start building our pipeline. So whenever you find that data set, that project, don't run and start uploading and running all kinds of pipelines. But this is just to show you that you don't need to download those pipelines or the samples first and create your pipeline. So after we do that process, I am going to go a little bit over time, but I hope you bear with me. So we download the SRA table, right? We upload it to the platform, and now we processed our data and we will have our table of expression. So how do we prepare that table of expression for what we want to do here? Here we will want to create a classification pipeline that will look at the different clinical subtypes that we know of, right? So these are going to be given to us in an annotation of the samples, and they're going to be things like basal and luminal and etc. And so we're going to take those abbreviations and train a model. And that model is then going to, first of all, predict what it can find in other samples that we did not give it as a training set. But also it's going to select for us the genes, the features, those classifiers that will help us identify similar types of samples in any other type of data. And so next time what we'll do is we'll try to apply the gene set that we found here to TCGA data. So what is supervised machine learning? Supervised machine learning, as you all know, the famous one is a classification of cats. So what if we take cats and dogs, right? And we provide some examples of pictures of cats and dogs, and we train some kind of a black box, an algorithm, to look at the differences. We're not going to tell us what to look at. We're not going to tell the model what to look at. We're just going to provide it with examples. And then we're going to provide it with a test set, something that it can test those predictions on, right? And we might want to have some confusing examples in our test set. So the model will take those samples, we will annotate it with class, and here you as the user do the annotation, so you can select whether you choose clinical subtypes, risk factors, or whatever else. And those are going to be called features. The genes are going to be called features. And then in our test data, we will provide something similar, but without that class annotation, right? And then we will apply multiple analyses to that data. So let's go ahead and do that together. Beepsha provided you with a link to some files for today's session. Test and train, yeah, the same. Well, okay, so later on, we will explore how to do that. Typically, you do a separation of 70% in your training data, about 30% in your test data. You do your classification, you're training your model and try to get it to a good accuracy. And then you will have a validation data set. That's what we'll do next time. For now, we have here two examples. So you can choose between the cancer cell lines that we're looking at now. So we have here already test and train data. Or this is the early data set for breast cancer, where again you have test and train. Okay, so you can use either one, but let's just go through a couple of examples to see how it works. And then next time we'll discuss the results. Okay, so we can use, you see here, we have, so test and train. What would be the difference? The difference is that in the train data, you see there's a class row. So let's take a look at this closely. Okay, so we have ID, these are cell line IDs, and then class, so normal like, basal, cloud and low, etc. And then we have a test. And so in the test one, there's not going to be a class and there's going to be different samples. So let's try this. Later on, we'll talk about the details. We'll go to areas of analysis, and let's go to supervised analysis and upload both of those data sets. So I forgot where I put those. How to make the training. So the training data set is going to be in the training data set is going to be in the class, right? So you just need to annotate them correctly. Sorry, this is the link. Let me download it because I forgot where exactly I put it. Okay, so the only difference is that for training data set, you provide examples. And then for the test data set, you eliminate that annotation, right? So you don't provide examples. Okay, so where's this? Okay. Okay, right there. So what we do is upload. I will upload the cell line data. So I'm just going to, for now, look at test and train right here. So I'll select both of them, upload them. How can we annotate based on what? Well, there is a supplementary table in this publication that tells us for each sample what clinical class it belongs to. And what we want to do is we want to create a list of genes that, based on RNA-seq data, will help us find the same molecular subtype. For now, this is an exercise. By the way, I really suggest looking at Transcriptomics 3, where all of this is explained in great detail, including preparation of the test and training data set specifically for this project. So please go ahead and check that out. It's going to be right here in the classification section. All right, so we uploaded our test and train data set. We just need to drag them in the appropriate group. So here we have the train and the test. Continue. And now let's select decision tree. No settings whatsoever. Just want to look at the output. So we'll have cell lines, test, train, decision tree. Okay, and I'll walk you some of the results, but please try and follow the Transcriptomics 3 that also provides you some of these data sets and provides some exercises so that you really understand how this works. So let's take a look at what the results would look like from running such a pipeline and explain what does it actually do. So the result from a decision trees are called binary decision trees. They're basically decisions that are binary, yes or no. Put it in this group or put it in that group. And it also provides you with a data source based on what? So threshold in what data. So here we see the result of a decision tree that separated everything into 3 groups: luminal, basal, and cloud and low. This separation is going to be based on the type of data that you put in. So it's going to be based on your training data set. If your training data set contains 4 groups, it will provide you separation to 4 groups. If it provides you 3 groups, it will separate in 3 groups, right? So you really have to think about the training when you are looking at the results. But how do we know based on what? So this is a gene, and it tells you the threshold. The threshold is below 4.25, right? What is below 425? Everything except for luminal, right? So here you have above 425, that's everything here, and that's all luminal. And below 425, which is all the rest of the subtypes. and then provide you another threshold to separate the other samples that we have, meaning basal and cloud and low. So here you have another gene, and here you can see everything above 5.2 is basal, everything below is cloud and low. So essentially, decision tree is like a process. Let me take everything and try to see which gene or which feature in my table can separate it into groups that I gave it. Now, if we do this based on both of these genes, it will look like a scatter plot, right? So in principal component analysis, we had PC1 and PC2. Here, we found two genes that together separate everything into three groups: group 1, group 2, group 3. And you can see that the samples are colored by type. Now you can do this multiple times and get different results. You can see that you can check those results based on annotation of these genes. So next time, we'll start looking at how to apply this to the full data set and think about how to validate such findings, right? So these two genes are called a gene set. So I suggest that we take a look at the paper that we started off with today that talks about the different cell lines and how they are validated in a clinical setting. Please take a look at Transcriptomics 3. We'll pick it up from here next time and continue talking about how the two data sources can be used together, one as a training and a test, and the second data set as a validation, and how to use multiple types of classification models to achieve the best results. So I'm sorry we went a little bit over time. I hope this was useful. Please let me Okay, so question, how to connect classification with drug selection? Excellent question. Here, specifically in the project of cell lines, we have a separate data set that talks about a high throughput testing of the different drugs. So if you go to the project itself, and the project is going to be, I was just looking at it, right? So the project could be found here. This one, Modeling Precision Medicine. So there you will find that we have a data set on both gene expression and on inhibition by different drugs. So if you can classify your samples using a table of genes and their expression, and then you can find a table of inhibition by different drugs, you can find which have a correlation, right? So if you can do that, then you can actually predict, right, which subtype based on molecular features will respond to different treatments based on high throughput screening of these different drugs. Can establish the relationship. Yes, exactly. So that is a challenge, as you will see. So we can set a time maybe to discuss this separately. I'll have to go in a couple of minutes, but we will be able to find some correlation after you use feature selection, right? So classification by itself is not going to be enough. You want to select for only a subset of genes and subset of drugs, and then find association between them. But maybe we can set up a time to discuss that in detail. Okay, another question. Normalization aspect of gene expression. Okay, so great question about normalization. Normalization is important as well as filtering, right? So there's a lot of noise in our data. The low expressed genes, the genes that are going to be non-informative or even confusing, and that's why I'm talking about feature selection or filtering. We have to account for that noise. And so normalization can help us reduce overall noise and maybe eliminate some technical variation. And then when we're more focused on detailed analysis, we can select and filter out the things that are relevant to what we are studying. Okay, so Mikhail, I'm going to send you an email with some description for correlation between drug and gene expression. And in our next session, we'll talk a little bit about that as well as looking at relationship between cell line data and data from TCGA. But in the meantime, if you could all please fill out the project submission forms so that we know which project you are going to be wanting to work on over the next few weeks. So if you could fill that out and we could help adjust our schedule so that we can, yes, Zainab, I will send it to you as well. We can schedule sessions according to groups. Perhaps we'll find several of you interested in the same topic, so we can actually make breakout sessions in the next few weeks and discuss your project ideas. All right, great. So looking forward to getting your feedback, and please work through Transcriptomics 3, some of the projects that we have that will help you get started, and then our discussions will be much more focused. So thanks again for joining today, and looking forward to seeing you next time.